I'd like to invite our next speaker, Professor Julien Calderero from the Henri Mondor, University Hospital Courtrai, just outside Paris. He's a liver pathologist who's been a leading force driving the use of AI in digital pathology. He's talking today more broadly about AI in clinical decision-making.
Thank you for this introduction and the invitation to discuss this topic, which is the application of AI to clinical data and what we can make of it. So you will see I will focus on imaging data because that's the field I think that is moving really forward so far. Okay. So basically what artificial intelligence tries to do is try to mimic what we do. So that was the definition initially. So try to automate things that human intelligence can do. And this was founded a while ago, as you can see. But I think that, of course, automation of different tasks is very nice, and to get rid of tedious tasks or tasks that takes a lot of time for physicians so they can do other things is very nice. But the most exciting thing I think about AI is try to do things so that we cannot do so far. So it does not fit with the initial definition, but really I think that the interest is try to extract information that we will not be able to extract without AI. And the true potential of AI is for very complex data, such as images like CT scans, MRI, or omics, or histological slides, as you will see. And it's less effective for common clinical data. If you have like a data set of, for example, 30 features such as age or size of the tumor, so far the conventional statistical methods still are very efficient, and the complex architectures that we use for AI does not do better, and even they perform worse. But for images, it's really useful, and the deep neural networks, which is one of the most used architecture for AI, really outperform any other methods for the analysis of images, because images are really complex data, because you have lots of pixels, you have intensity values, and you have the spatial relationships between the pixels. So you have a lot of information that is impossible to analyze extensively with conventional methods. Here are some examples of AI applied to imaging, and then after I will focus on histology. So there have been very few papers on cholangiocarcinoma because of the rarity of the disease, and you will see that when we apply AI, the more cases we get and the more data we get, the better the model will be. So that's a critical point, and I think that's one of the reasons that there are not many studies about cholangiocarcinoma so far, but that was a study that aimed, for example, to predict recurrence after resection of peri-hilar cholangiocarcinoma. So what they did is that they took imaging from a patient with peri-hilar cholangiocarcinoma, and then they trained a model, and this was able to tell if this patient will experience early recurrence or late recurrence. Another study on CT scan where they took the CT scan of patients with intrahepatic cholangiocarcinoma, and they tried to predict the recurrence, the early postoperative recurrence, by processing the CT scans that were performed before surgery. So again, one major issue with cholangiocarcinoma so far in the study is that there is no completely large validation data set, because one of the problems that we have with AI also is that when you train your model within your center, the model, of course, will detect features that are associated with the disease aggressiveness or response to treatment. But the main problem is that it will also detect features that are associated with your center, for example. And when you will apply the model to another center, to other hospitals, other patients, this will not work. So that's what we call overfitting, because you have a model that is really highly accurate for your patient, but then you will not be able to apply it to other patients. And for that, what we need is very large data set, heterogeneous data set from various hospitals that with CT scan performed with different vendors, different machines, in order to get rid of this very common phenomenon that we call overfitting. So now I will focus on pathology because that's the field that I know best. So initially, radiology and imaging medicine were really ahead of us in application of AI because they were digital largely before us. But now, since approximately 10 years, we have the ability to transform the slides that we see in pathology labs into digital files. So we are still late because most of the labs so far do not have this complete digital workflow because it's expensive in terms of IT storage of slides. And so far, the administration of the hospitals do not see really what benefit they will have in investing in this technology. So that was the answer they gave us first when we tried to convince us, for example, that we need digital pathology in our department. And they said, okay, but how many jobs will you give back? And we explained them, no, it's a step more. So you still have to produce the slides, then you have to scan it. So it will not allow to have less people in the lab, and likely we'll need maybe more people in the lab. But now they are understanding that it's the new standard, that everyone now is getting digital, and if we want to be competitive and to attract people to work in the hospital, we need to be digital. So what we do when we transform a slide, we put this slide on this the particular device, and the slide is transformed into a complex image file, and this image file can be viewed on a computer, and you can zoom in, zoom out, just like with a conventional microscope. And just some slide on how it works, and it's true for all images. It's not only pathology slides, it's true also for social network, where they can tag your face automatically in a picture, for example. So to understand how it works, you have to remember that an image is basically a matrix of numbers. You have the pixel intensity values, and you have the X and Y coordinates of each pixels. For pathology, the particularity is that we have color images. So these images are encoded in RGB, which is a very common way to encode images. And for one image, you have three matrices. One matrix is for the red channel, one matrix for the green channel, and one matrix for the blue channel. And the final image is the superposition of all these three matrices. What we do when we try to apply AI to pathology, first we have to cut the slide, because one slide is between 2 and 4 gigabytes. So it's a huge file, and the networks, the AI networks that we have so far are not able to process this huge file. Maybe in the future this will be possible, but so far it is not. And so I told you that there is huge cost for the storage of slides. For example, in my lab, we produce approximately 1,300 slides per day. Then you multiply this by 2 or 4 gigabytes, and you have an idea of how many tera of data we produce each day and that we have to store. So what we do, as these slides are far too large to be processed as is that we do what we call tiling, that we cut these images into small patches that are approximately 250 pixels. And then how does it work? The process of training a model. So first you have to have your image, and you have to have the label that you want to predict. It can be a mutation, it can be a transcriptomic subgroup, it can be the age of the patient, or it can be the outcome, recurrence or no recurrence. So let's say we want to try a model that will recognize if the tile that we give is a tumor or is non-tumor liver. So you give, for example, a first tile of a tumor, you give the label, you say to the model, this tile contains tumor. The first step of the model will be to extract numerical values, and then these values will be processed in complex mathematical layers, which are called, and each nodes that you see here is called a perceptron, and it is supposed to mimic how the human brain works, and that's why they are called neural networks. And the model will see the patterns in the features, the mathematical feature it extracts that are related to the feature you want to predict. And then you have an output value, which is usually between 0 and 1, because it's the probability of the image to be tumor, for example, or not tumor. And let's say we give a first image, we say it's tumor, then you have an output value of 0.8, so that's good, it's closer to 1, so the model seems to make a good prediction. The second image is non-tumor, you have a value of 0.2, again it's fine, it's closer to 0, and the actual image is not tumor. Then you give a third image and you have a value of 0.4, so that's not good because we want the probability to be closer to 1. And what happens is that each of the layer of this mathematical layers will transform themselves in order to change this value that is 0.4 and to bring it closer to 1. And of course, you do not want to change too much the values that were correctly classified. So with that, we can then try to do things, as I said, that we cannot do by the eye. Of course, you can automate diagnosis if you want. Like, it works very well for prostate cancer, for example. There are already products on the market that are able to reach like pathological level for diagnosis of prostate cancer on prostate biopsy. But you can try to do things that we cannot do so far. And this is an example of a project that was performed in collaboration with the team of Jacob Katter and Narmin Ghaffari in Germany, where we focused our work on a particular subtype of liver cancer, which is combined hepatocellular cholangiocarcinoma. So, you know, in the liver, the two primary most frequent liver cancer are HCC, hepatocellular carcinoma, and cholangiocarcinoma. And there is this entity that has features of both HCC and iCCA, and clinicians usually do not know what to do with this patient, what treatment we should give them. Should we give them treatment for HCC or treatment for cholangiocarcinoma? So there is high quality evidence for HCC, iCCA, but not for these combined cases. And interestingly, when you analyze molecularly these tumors, you can see that some of them, and significant subset, really fit into the HCC group on transcriptomic analysis, and some of them fit with iCCA on transcriptomic analysis also, and the alteration are sometimes those of HCC and sometimes those of iCCA. So what we try to do is we try to determine if AI could reclassify these tumors that are very rare and that are very difficult to treat as HCC or iCCA, because this has a huge implication in terms of prognosis and also systemic therapies. So we built a model by giving the model slides of HCC, and slide of iCCA, like 500 slides of each. And we were able to first develop a model that was able to differentiate HCC versus iCCA with a very high performance. And you have here some example of slides of conventional HCC and conventional iCCA that were predicted, and you have the color scales that depend on the diagnosis. And you can see on the upper right that the blue area is considered HCC, and that's fine because this is an actual HCC, and here it's in red, and this is an iCCA. Then we apply this classification to combined cases, and we saw that the classification had a prognostic relevance because patients with tumors that were reclassified as iCCA had a worse outcome after resection or after liver transplantation. And we compared this AI reclassification with a simple assessment by the eye with the most predominant contingent, and the conventional pathological analysis did not have any prognostic impact. Then we try to go deeper and to see if the classification has a molecular relevance, and we sequence a subset of cases. And we then looked at the alteration that was specific for HCC versus iCCA. And you can see here that the classification is not perfect, but still, for example, cases that are alterations that are more frequent in HCC were all reclassified as HCC by the model, and also molecular alteration more frequent in iCCA were more frequently reclassified. Tumors with this alteration were more frequently reclassified as iCCA. And what we can do also, that the technology allows us to do, is combine all these approaches with the new omics technologies. And there is now the rise of what we call spatial omics. And basically what you can do with this technology is have the gene expression, but in situ, in a tissue section. And you have an example of how it works. You place some tissue section on a particular slide. You have many spots, like 1,000 spots on these slides, and you have the gene expression of each of these spots. So it allows you to have an idea of how the gene expression is in situ and in every area of the tissue section that you placed on this particular slide. And for example, this is a liver cancer, and what you can do is then perform clustering on all these gene expression spots. And you can see here, that was an example. We just perform an unsupervised clustering. And you can see that you identify distinct tumor areas that have distinct gene expression profile. Some are related to stroma, some are related to differentiation. And you can have an idea of the intra-tumor heterogeneity. What we did in this study, and again, in combined cases, is that we investigated the relationship between the AI models and the spatial relationships. Because what is nice also with these AI models is you can have prediction heat maps on the slide that will tell you which area impacted the classification. And you have here the example of a combined HCC-CCA. This is the H&E section, the common H&E section, and you have the prediction heat map of the model, and basically the area in red for the model is reclassified as iCCA. Then what we did is try to understand how the model worked, because that's a critical point, and there is this hampers the use of AI in clinical practices, because it's very difficult to know how the models work. I think to be implemented and to gain the trust of physicians, it will be mandatory to have at least an idea of how the models work, at least partly. So we did spatial transcriptomics, and you have some example here of genes that are related to biliary differentiation, like keratin 7, and you see that it grossly matches the area that is considered as iCCA. And this is albumin, which is more expressed in areas that are predicted as HCC. Next, so besides diagnosis, what you can do also is predict prognosis. And so there are no studies so far on cholangiocarcinoma with true validation set that predict prognosis from digital slides, but this is a work that we did in HCC, and basically what we saw is that when you process slides of the patient with hepatocellular carcinoma that was treated, who was treated with a surgical resection, when you process the slide, you can predict the prognosis and the outcome of the patient, and better than a score that will combine all conventional features. And finally, and I think this is probably one of the most interesting thing, is prediction of response, and also, as we saw, response to chemotherapy, but also response to immunotherapy. You know that the field is changing for cholangiocarcinoma with the combination of immunotherapy and chemotherapy. So it will become critical to identify the patient that really benefits from this particular treatment regimens. And again, you can apply AI to slides in order to predict gene signatures that are relatively difficult to implement in clinical practice, because you need to have a molecular biology facility, and there are also standardization issues. And that explains that so far, for example, apart from breast cancer, there is no really gene signature that is widely used in clinical practice. And what we did in this work is that we classified samples of hepatocellular carcinoma according to different gene signature that may be associated to response to immunotherapy. And then again, we trained a model that was able to predict them with AUC that were above 0.8 in most of the cases. And what is important also to know is that you can modify the thresholds of your model if you want to have a highly sensitive or highly specific models, because it's not likely that the model will be really perfect and will completely rule out molecular biology, but you can try to use this model to do some kind of triage and, for example, exclude some cases for which you are sure that they do not show activation of the signature, for example. Again, you can then map the prediction that you obtain with the spatial transcriptomic, and that was one case of hepatocellular carcinoma for which we have the prediction map here and the expression of the gene signature. So it's not perfect, but you can see that it grossly matches the same area. So I would like to end with this slide, which is what we call the Gartner curve, which is applied for every new technology that comes out. And you have the time on the y-axis and the visibility and the hype on the y-axis and the time on the x-axis, sorry. So when the technology begins, you have this really high peak of inflated expectation. Everybody thinks that this will solve everything. Then we begin to see the limits of the different systems, and you go through the disillusionment, sorry. And then you learn from the errors, you adapt the technology to get rid of most of the problem and the errors. And then you will reach what is called the plateau of productivity, which are really, for AI, the clinical expectations. So I think now we are maybe here, or so it depends on some people. Some people are still here, and some people have understand that we will face some problem for implementation. And but I think we are like here. We begin to have products that are commercially available for some simple task. And now the challenge will be to bring this plateau of productivity as high as we can. And again, also, I think what is key for that and for all the kind of models that I presented for prediction of response is to try to have access to samples from clinical trials. And you know, when you publish, for example, a study with gene sequencing, it's mandatory to share your data. And there is a movement also in the industry to try to make the data from clinical trials available after the publication of the trials. Because, for example, if you want to implement this kind of model, you need really good quality data, lots of data, and it's very hard to do that retrospectively. You have to gather as much data as you can. So I think that will be key, really, to implement this kind of models in clinical practice. But however, it's really complex data to obtain. So I would like to end by thanking all the people that are involved in the different work that I mentioned, and also I'd like to say that if you know someone that would be interesting to work on this kind of approaches, we are hiring a postdoc in the lab, and you can just send me an email. Thank you.